everybody. Welcome to this month's iteration of the Innovate seminar. Today we have Trevor GrandPre, who is a postdoctoral fellow at the Center for the Physics of Biological Function at Princeton University, and who is also a Schmidt Science Fellow. Trevor is an alumnus of DePaul University, where he did his undergraduate degree, and UC Berkeley, where he did his graduate work and earned his PhD in 2021. And since 2021, he's been at Princeton as a part of the CPBF. And today he's going to give a talk on his work on the effects of linker length in biomolecular condensates. So I think without further ado, I'll pass it over to Trevor.
Thank you, Carlton. So as mentioned just a second ago, I'll be talking about effects of linker length in biomolecular condensates. And there's a lot of jargon here, so I'll break it down over the course of the talk. Here's a snapshot, or a video actually, of a simulation that's modeling a specific biomolecular condensate. And so I'll talk to you more about this model, and also I'll talk to you about the motivations for why we care to model it. Okay, so before I jump into this, I would like to give a broad introduction to what the overarching field of membraneless organelles or biomolecular condensates. So here's a picture of just a generic cell. More it looks like an animal cell, and all the little blue, blue. I don't know all the names of these colors, but all the different colors here in circular shapes are so-called membraneless organelles. So they don't have something that's keeping them together, a membrane that's keeping all the proteins together. So for instance, let's look at the nucleolus within the nucleus. This is something that has no membrane, and it's just a whole bunch of interacting nucleotides that are keeping them compact. And so the same thing for the other droplets here. And so one definition is just biomolecular condensates are concentrated biomolecules in small volumes without membranes. But there is, some controversy with this, or it's not fully understood which proteins or DNA or RNA in the cell is this membraneless organelle or biomolecular condensates. But a simple picture that has an equilibrium picture that has emerged is, well, how can so many of these droplets, like within the nucleus, this PML body and the nucleolus and the polycomb body, all of these, how can they all, be confined but not mixed? Because if this was an equilibrium system, we'd expect all of it to be mixed into one big mixture of all the proteins. And so one simple theory is, well, these are all, you can think of it as some like multi-component phase diagram where I can have multiple protein concentrations that are in equilibrium with each other. Of course, this is a simplified picture, but this is just one idea. And of course, these multivalent proteins. You can think of these, the red and the blue. The red and the green as, for instance, multivalent proteins, where there's regions, so-called sticker regions, where these are regions that have a high affinity to, stick or bond to other green proteins, for instance. And there's a linker region, which is, in this framework, it's just flexible. It's just a flexible unit that allows the stickers to, move around to bind to other stickers. And I guess one last thing is that within this, so this is the framework that I'll consider this so-called sticker space or sticker linker framework. But one thing to notice here is that membraneless organelles, I guess the point is that, each one of these has a biological function, and membraneless organelles imply a biological function, but biomolecular condensates may not. They might, for instance, be caused by accident or by a side effect of another process. So I guess that's also a big part of this field, is understanding, well, do some of these biomolecular condensates have a biological function, and how much is the dynamics and microscopic interactions between these proteins give rise to their function. Okay, so that was sort of the overarching idea, but now let's dig more into the sticker spacer idea as brought out in the green polymer. So between the stickers is what I called was the linker, and actually the linker itself has can change the properties of phase separation, or can affect phase separation. So one example is that within a protein called LAF1, so LAF1 is an intrinsically disordered protein within P granules. So that's a lot as well. So LAF1, intrinsically disordered region, it's a region with so-called low complexity. It has a lot of repeating units, but it also has it doesn't seem like it has function, but of course we know it does have some function. But anyway, this is just to say that some linkers are typically intrinsically disordered. So anyway, and P granules are within frogs, frog embryos, and they are sex organs, so they're important whether they become sperm or egg in frogs. But the idea here is that this linker length within this LAF1 protein, which is on the left is the wild type, you see it's concentrated within, into these bright spots, right? But if you delete the intrinsically disordered region, you see that the phase separation is now gone. It's kind of spread out. The white is all spread out. And so this is one idea of saying that the linker properties are important for phase separation. Another important point is here in VRN1, this is a protein that is important for flowering, repression of flowering in Arabidopsis. And so in this system, they artificially sort of change the number of proline serine repeats within the linker region of VRN1. And so for 100 proline serine repeats, you see that there's you see these red droplets, which is the phase separation. But if you make it longer, so if you make it 120 repeats, then you see the phase separation goes away. So this is just to highlight where I'm going with this in my talk, which is that linkers have several can affect these biomolecular condensates in several ways. One is by occupying volume in condensates, and another is by adding interactions. So for instance, there could be charge patterning. They could be charged, or by linking, keeping stickers together as in a polymer form. Another thing is that they can exclude intruders. So as I mentioned, like in the previous slide, for instance, there could be red proteins and green proteins that are separate. There's not red proteins in the green. So this is another way to, keep things separate, as the linkers can help with this. And lastly is there can be an interplay with relevant length scales. And what I mean by that is, if you have a rigid component, like a rigid enzyme, and that has stickers at specific lengths, then this will give rise to phase separation properties when interacting with like a flexible protein. And so this is, I'm not sure if that one made complete sense, but this is the idea that I'm going to emphasize in my talk. So we'll talk more about this. This last point. Is there any questions so far?
None at the moment.
Okay.
But we can cover all questions at the end. Yeah.
Okay, sounds good. Okay, so in my particular study, there's something called the pyrenoid, and so the pyrenoid is a membraneless organelle within a green algae called reinhardtii. And so within the chloroplast, there's this pyrenoid that its purpose is to concentrate carbon dioxide to then convert it into sugars for photosynthesis. So the pyrenoid has, so this is showing you how dynamic it is. So here the colors are different, but the green is the pyrenoid, and you see it dissolving and reforming into 4 daughter cells here. And so it's a very dynamic process, and showing that it's sort of liquid-like in nature a little bit, where it can dissolve and kind of reform new cells. So that's one property, is that this thing is dynamic. But also, if you can reconstitute the pyrenoid and see phase separation in vitro, which has 2 main components. One is Rubisco, and so Rubisco is a rigid enzyme shown on the top here, and it has 8 binding sites or stickers for EPYC1 shown on the top in the blue and purple, and on the bottom. And you may have heard of Rubisco. It's actually the most abundant enzyme on Earth. So like 97% of organisms that do photosynthesis have Rubisco. But the mechanism for increasing the rate of carbon capture is different. For instance, in land plants, they have their own mechanism, and green algae has their own mechanism. And yes, this is a necessary step that has, each set of organisms or species have found to increase the rate of carbon capture from Rubisco, because it's not very efficient by itself. And EPYC1 is, I'm showing here, so EPYC1 is an intrinsically disordered region that has, protein that has 5 stickers for Rubisco. And so the 5 sticker regions, so I'm showing here the amino acids that make up EPYC1. And so the 5 highlighted, the yellow highlighted regions are the sticker regions that will stick to Rubisco, and the other regions are the linkers. And so you can't visualize an intrinsically disordered protein in the structure, how it folds and things like that. So I'm just showing the sequence. But okay. So with these 2 components, you can see that, because Rubisco is a rigid enzyme and EPYC1 is flexible, that there can be properties of the EPYC1 linker length that might affect phase separation in a non-trivial way. And so that's what I'm going to address here, is how does EPYC1 linker length affect condensation? Okay. And so here's actually an image of 3 Rubisco, I've been saying Rubisco, but these like subunits here are actually hollow enzymes of a larger structure. But anyway, so this is showing that 3 Rubiscos are being held together by EPYC1s, where this is from cryo-electron tomography. But one thing to note is that the linker lengths are interpolated, so they're to be the correct length, but they're not like from an experiment. They're like artificially drawn in. But the sticker regions are true. So the where you see like the orange, or is this red or orange, one of the 2 on the stickers. So that is EPYC1 sticker region being bound to Rubisco. But this is just one way how Rubisco can non-trivially pack to create sort of larger, what will be a big part of the pyrenoid that I showed you earlier. So with this, 2 things that I get asked a lot. One is, well, why do I care about green algae? And that's a good point. Other than just the sake of understanding what's going on here, there is actually a biotech application. So for instance, there's groups here and also in the UK who are trying to engineer pyrenoids into land plants, such as corn and things like this, to increase the yield in air and to be able to grow in areas where there's not a lot of sunlight. So this is one avenue where modeling could provide insight into like these being able to provide more food, but also because Rubisco, the pyrenoid is good at carbon capture. I mean, there are also people who are trying to use this to combat the carbon problem that's around. So with that, let me jump into my model. So the model is, Rubisco here has is on the left. So it has a 10 nanometer base and a 2 nanometer stickers that are at 45 degrees from the top and bottom. And this is just based on the crystal structure that I showed you. And EPYC1 has 5 stickers that will bind to the Rubisco stickers, and the linker length is set by the spring constant K. So larger spring constant means that the linker length will be smaller. Smaller spring constant means larger linker length. And there's excluded volume between all elements except the cyan and the stickers, pretty much. The sticker interactions okay, but between Rubisco and EPYC1, but everything else.
Brownian dynamics, statistical mechanics. Yuck.
And so, but all this is together to provide one-to-one bonding between all the elements, and the stickers interact with this potential U-knot. So here, I guess the main point is that molecular interactions between this Rubisco and EPYC1 involve 2 microscopic parameters. One is the spring constant here, K, that tunes the linker length, and a U-knot, which tunes how strong the stickiness between the 2 components are. And so because in experiments, this is not something that can be experimentally tuned, so we wanted to now understand how these 2 components affect Rubisco EPYC1 binding through the dissociation constant, because that's one thing that can be measured experimentally. And so here's a little simulation showing one EPYC1 and one Rubisco, and you'll see that a binding event will happen now, and then it'll eventually fall off sometime. Anyway, yeah, okay, you don't see it fall off, but I promise you it falls off. And the idea is that, as shown in this equation in the bottom right, you can take this E in brackets as the concentration of EPYC1, and KD is the dissociation constant, and PB is the probability of EPYC1 being bound to Rubisco. And so from computing the fraction of time that EPYC1 is bound to Rubisco, we can actually back out the dissociation constant as shown in this plot in the bottom right. And what you see is that the linker length does have a large effect on dissociation constant. So here, a larger spring constant K equals 1.2 is sort of like the short linker, and the K equals 0.1 is the long linker. And so in general, the longer linkers, or this red, has a stronger dissociation constant, meaning it's lower. The lower the dissociation constant, the stronger. And that's pretty much because it, as shown in the left plot here, is that it can access more stickers of Rubisco. A single EPYC1 sort of has to stretch to access other sites of the Rubisco, and because the longer linkers can stretch farther, they can access, a third binding site, for instance, shown in the plot on the left. And 2 pictures of this. Oh, so experiments are down here. So there actually my experimental colleagues here have measured the dissociation constant, and it's about 30 nanomolars as an upper bound. So this shows that we can parameterize this specific, microscopic parameters to experimental parameters through this dissociation constant. And just here's a little picture of what's going on. So for the larger spring, you see that the linkers are so short that you kind of can't bind consecutive stickers of EPYC1. You kind of have to skip one to be able to bind a consecutive Rubisco sticker. But for the smaller spring, or the longer linker length, you can almost fully, bonds several more spots and bind consecutive EPYC1 stickers to consecutive Rubisco stickers. And so in principle, I guess, okay, I showed you molecular bonding, there's association constant, but do these dissociation values lead to condensation at realistic concentrations? So that's what I'll go into now. So here I'm going to show you two simulations. The top one is going to be for the short linker system, or the large spring constant, and the bottom will be for the long linker system. And so let me know if you see any difference. Now, of course you see a difference, but I guess the question is why? Why is there such a big? Why is just changing the linker length produce such a big difference in the phase separation properties? Where here the short linkers actually phase separate well. You see a big slab here held together, but for the long linkers, it's getting ready to disintegrate. You know, it's sort of on the verge of being dissolved. So to understand this from the microscopic level, I want to actually, I did some microscopic measurements of the dense and dilute phases and to try to understand the molecular mechanism for why linker length has this effect, such a large effect. So first, on the dense phase, what I found is that if you look at the bonded stickers per Rubisco in the dense phase, so I mean, it's in that slab or condensate, and you just measure how many stickers of a Rubisco are bound or by an EPYC1 sticker. So in the two systems, the long and short linkers, there's no difference. It seems like the distributions are the same, but okay. But where there is a difference is now if I take the same, if I look at a particular Rubisco in the dense phase and I ask, well, how many different EPYC1 molecules are bound to Rubisco in the dense phase? Now we see a difference where the long linkers are associating with about 4 or 5 different EPYC1s, but the short linkers are actually associating with almost 7 or 8 different EPYC1s. So this gives us the idea that long linkers allow multiple stickers of an EPYC1 to be bound to a Rubisco, single Rubisco. And so you have these two pictures where on the left is the short linkers where you almost have like a hairball image where it's like almost every sticker of Rubisco has one EPYC1. But on the right, which is the picture of the long linkers, you see that in the dense phase, it's almost fully bound by a smaller number of EPYC1s. And okay, so now I want to also look at the dilute phase to compare and contrast with the dense phase. So in the dilute phase, the dilute phase here is just everything that's not the condensate. And so what you see is that if I look at in the dilute phase, if I measure the bonded stickers per Rubisco in the dilute phase, the short linkers are not able to bind all this. So in the short linker system, Rubisco are not satisfying all of their bonds in the dilute phase. However, in the long linker system, they are able to satisfy all of their bonds. Right. But if you look at the molecular partners per Rubisco, how many different EPYC1s are bound to a specific Rubisco. Now you see that they're the same. And so this shows you that these short EPYC1s linker systems can't span between adjacent Rubisco binding sites. And so what this means is that there are Rubisco so for the short linker system on the left, there are Rubisco that have stickers available. And so it's thermodynamically unfavorable for them to sort of stay out in the dilute phase when they can join the dense phase and satisfy all of their bonds. However, the long linker system on the right, once again, can satisfy all their bonds with a small number of EPYCs, so they're kind of happy being in the dilute phase. And so this is one reason why this is just showing us from the microscopic level why the long linker system would have a weaker phase separation than a short linker system. And we can also look at this from the perspective of the thermodynamic theory. And so because that was more of sort of a microscopic, measurements, but it's not really a full theory. So to really understand what's going on here, I constructed a theory called the so-called dimer gel theory, where you define a free energy density, which has three terms. One is just the, what is this? Sorry. Okay, at least that works. Where the non-interacting term is just the ideal contribution, or just the entropy of mixing, if that makes sense. And then the middle term is just the excluded volume. So here we're modeling it as hard spheres. So this is just a new R is just the volume, excluded volume for Rubisco. And new E is for EPYC1 of a sticker, so the effective sticker volume. And so this is just a hard sphere approximation. So obviously they'd be kind of squishy, but here we're just approximating them as hard spheres to get a qualitative understanding. And then the specific interactions, or how EPYC1 is associating with Rubisco, we use this term here, where it's a minimization between a dimer free energy and an independent free energy. So what are these two? I'll break it down. So the dimer free energy is the free energy to form molecular dimers, or where a Rubisco molecule will bind to an EPYC1 molecule. But the independent contribution is the free energy to form independent bonds between just stickers of Rubisco and just stickers of EPYC1. And the reason we do this minimization is because we found that you can compute it in both phases, like at the dimer free energy and independent free energy, but we found that the dilute phase is actually dominated by the dimer free energy, the one on the left, and the dense phase is dominated by the one on the right, the independent. Okay, so with all this together, we can actually produce, we want to make a prediction to see if it agrees with what the simulations were showing us. And so here's, here this is a prediction of the phase diagram, where KD is the dissociation constants for the short and long linkers. So the blue is for the short linkers and the red is for the long linkers. And the KB is a fit to the simulations to get the right dilute phase as in the simulations. And so from this, we see that, okay, as you can see, the short linkers have a more dilute phase. And then when you look out to the full phase diagram, this X here is where my simulations were done. And we see that it confirms this idea where the blue, or the short linker system, has a larger phase diagram, and it's also more dense because it's more to the right in molecular concentration than the red, which is the long linker system. So at least it's qualitatively agrees with this, free energy picture of why the short linkers can phase separate better. So I think I'll just go to the general idea now, where what we found is that EPYC1 linker length controls phase separation via a molecular fit between EPYC1 and Rubisco. So this is sort of the diagram of it. So if you have a poor fit, as shown in red, then you're not able to, fully bond, one molecule of EPYC1 is not able to fully bond to Rubisco, and so this leads to a larger phase diagram. If you do have a good fit, then, between these two molecular components, then it's a smaller phase diagram. And so in essence, molecular fit is inversely proportional to phase separation. And it depends on these relevant length scales of the Rubisco binding site spacing compared to EPYC1 linker length. So for the sake of time, there are other implications that I will not go through in detail, but the idea is that there's pyrenoids in other organisms, not just green algae. There's pyrenoids in carboxysomes, and they have stickers in different regions and linkers at different lengths. And it seems to qualitatively agree with the similar with the framework, I said that there's a mismatch between linker length and Rubisco sticker spacing that leads to condensation. And these are just sort of other avenues that I also won't go into. But the idea is that, these pyrenoids also have tubules. And so this is, where things are going, where, you can imagine a pyrenoid wetting a tubule, and there's interesting statistical mechanics to learn here too about how linker length and other microscopic properties can affect wetting. Okay, so with that, I'd just like to conclude and say that linker length relative to an intrinsic length scale can control phase separation. And in the pyrenoid model, short EPYC1 linkers can't span Rubisco-binding sites, but can connect Rubiscos in dense phase and drive phase separation. And I'd like to thank my collaborators and also my funding sources. And I'm happy to take questions.
Thank you, Trevor, so much for the great talk. I just want to remind all of the attendants that you can submit questions using the Q&A feature at the bottom of the panel. But I did have a couple of questions of my own. And I think the first one is sort of like a methods question. And how did you, in your simulations, take measurements between the dilute versus condensed phase? So is this like the equivalent of changing the temperature of your system?
Oh, yeah, that's a good point. So here I actually measured so I took a time integrated density profile along the x-axis here, the long axis, and from that time integrated profile, I sort of shift the slab to be in the middle to get sort of an invariant profile, and I sort of cut it off where when the condensate is, or the density of the profile is peaked.
Okay.
It reaches like its level, this plateau value.
Yeah. I think other question I had was I know that I think you had said earlier that there's like some difficulty experimentally measuring linker length, or not measuring linker length, but for controlling for linker length. Is like linker length, I guess, like an analog for, I know that it's an analog for the stiffness between like the two stickers on EPYC, but is there like any like thoughts on how you would be able to experimentally control for linker length? So like
in this system,
some way affecting like the structural integrity, or with like a genomic edit or whatever?
yeah, well, yeah, for this system particularly, we have been thinking about this. It's something that is sort of not being done at the moment, but it is something that, for instance, we've tried, you can just do like repeats of, I think we've tried repeats of different amino acids, and we found some candidates where the folding, using like AlphaFold, we found that the fold was it made the same protein as EPYC, the native EPYC1, by just adding a repeat of specific amino acids. I can't remember which exactly what the repeats were, but yeah, we have some candidates for how to do this without affecting, the function, how the protein functions. no need to to force any questions.
Oh, I guess I had one more question.
Mhm.
And I think maybe if we can do closing remarks. But I think this last question is somewhat broad. But I remember earlier in the talk you had mentioned that Rubisco is like commonly seen in like multiple uh uh photosynthetic organisms, and that the sort of like the stickiness between Rubisco and EPYC like serves like some functional purpose. But I guess I was wondering if you sort of see like differences in phase behavior that depend on the exact like a mechanism that Rubisco is used in. So, for example, like say in bacteria A, you do see this like phase separation behavior, and it has like a very like well understood mechanism for how like the organism is able to undergo photosynthesis. But for another organism in which you maybe see a different kind of a different phase behavior, would you also, I guess, see like a different mechanism for like carrying out photosynthesis?
Oh, I didn't get all of that, but I think you're saying like, does the structure of the condensate, the pyrenoid, affect the function? I think it's boiling down to this, but I'm not, maybe because what I'm saying is like each Rubisco. Okay, so I guess there are functional requirements, right? So like a pyrenoid needs to be able to not be too dense because there needs to be there's other proteins that are important and they have other important jobs to do. So you don't want it to be too dense. But I guess, and I think, implicitly, like, yeah, there's more going on than just, the pyrenoid itself. There are, I didn't tell you about the carbon concentrating mechanism. So there's more to it, but I think that how dense and how it forms is important in the biological function, because some pyrenoids are more solid-like, and that's definitely some contribution between the interactions between the Rubisco and other proteins. But yeah, there's more going on. This was just part of it. I don't know if I did a good job.
Oh no, this I think this more than answered my question. Thank you.
Okay.
So I think with that being said, I'd like to thank you for giving a great presentation today. And I'd like to thank our contacts at KITP and the broader NSBP executive and student council who've helped set this up. I think this was a really great talk. Thank you.
Thanks. Thanks for inviting me.
Yeah.
Thank you, Trevor.